I'm Dr. Amanda Toland, Associate Professor of Cancer Biology and Genetics at The Ohio State University Comprehensive Cancer Center, James Cancer Hospital, and Solove Research Institute. Cancer researchers learn a great deal about healthy cells and cancer cells by preserving them, staining them in different ways, and then studying them under different kinds of microscopes. Here are a few examples of cells that have been prepared and stained in 3 different ways to bring out particular features. Here's an example of light microscopy. This is a section of lung tissue that has been sliced very thin and a tumor that was growing inside a human lung. The tissue is stained so that the nucleus of each cell is colored dark blue and the cytoplasm is pink. The large round mass is the tumor. The white areas are air passages in the lung. This image was taken at a low magnification of 4 times. Here is an image of the lung tumor taken at a magnification of 400 times. It shows more of the cellular features of the tumor. Each darker blue circle is a cell nucleus. The nucleus of each cell is stained blue. The cytoplasm is stained pink. The air passages are white, and these are cancer cells. Next are examples of immunofluorescent microscopy. The cells have been stained with dyes that emit light of different colors when exposed to ultraviolet light, or UV. The cells are viewed through special UV microscopes. The image is also of a lung tumor from a patient photographed at much higher magnification. In this image, the blue color is cell nuclei. The green, or yellowish color, shows the presence of protein molecules for a particular signaling pathway that transmits signals from the cell surface to the nucleus. This second image shows a lung tumor from a different patient that was stained in the same way. The blue nuclei are visible, but the green stain is absent. This means that the particular cell signaling pathway is not activated in this patient's tumor. Whether the signaling pathway is active, as in the previous photo, or not active, as in this photo, can help determine the best treatment for certain lung cancer patients. The third type of microscopy is electron microscopy. It uses a stream of electrons rather than light to view the cells. The cells are usually stained with gold or a metal that is electron dense. Cells can be viewed at much higher magnification and resolution this way. This image shows a cell from a lymphoid tumor. The nucleus is dark and clearly visible, along with mitochondria, the power-producing organelles of the cell. This image is one of even greater magnification, showing details of individual mitochondria. One difference between normal cells and cancer cells is that cancer cells have many more mitochondria. We've reviewed 3 types of microscopes that enable us to view cells in a variety of ways, advancing our understanding of cancer.